Nipah virus, scientific name Nipah henipavirus, is a bat-borne virus that causes Nipah virus infection in humans and other animals, a disease with a high mortality rate. Numerous disease outbreaks caused by Nipah virus have occurred in South and Southeast Asia. Nipah virus belongs to the genus Henipavirus along with the Hendra virus, which has also caused disease outbreaks. Like other henipaviruses, the Nipah virus genome is a single, non-segmented, negative-sense, single-stranded RNA of over 18 kb, which is substantially longer than that of other paramyxoviruses. The enveloped virus particles are variable in shape, and can be filamentous or spherical. They contain a helical nucleocapsid. 6 structural proteins are generated: N, nucleocapsid; P, phosphoprotein; M, matrix; F, fusion; G, glycoprotein; and L, RNA polymerase. The P open reading frame also encodes 3 non-structural proteins, C, V, and W. There are 2 envelope glycoproteins. The G glycoprotein assembles as a tetramer to form the viral antireceptor or attachment protein, which binds to the receptor on the host cell. The F glycoprotein forms a trimer, which mediates membrane fusion. Ephrins B2 and B3 have been identified as the main receptors for Nipah virus. Ephrin subtypes have a complex distribution of expression throughout the body, where the B3 is noted to have particularly high expression in some forebrain subregions. The most likely origin of this virus was in 1947, 95% credible interval, 1888 to 1988. There are 2 clades of this virus, one with its origin in 1995, 95% credible interval, 1985 to 2002, and a second with its origin in 1985, 95% credible interval, 1971 to 1996. The mutation rate was estimated to be 6.5 times 10^4 substitution/site/year, 95% credible interval, 2.3 times 10^4 1.18 times 10^3, similar to other RNA viruses. Nipah virus has been isolated from Lyell's flying fox, Pteropus lyellii, in Cambodia, and viral RNA found in urine and saliva from P. lyellii and Horsfield's roundleaf bat, Hippocyteros larvatus, in Thailand. Infective virus has also been isolated from environmental samples of bat urine and partially eaten fruit in Malaysia. 8. Antibodies to henipaviruses have also been found in fruit bats in Madagascar, Pteropus rufus, Eidolon diprenum. 9. And Ghana, Eidolon helvum. 10. Indicating a wide geographic distribution of the viruses. No infection of humans or other species have been observed in Cambodia, Thailand, or Africa as of May 2018. Fever, headache, muscle pain, vomiting, sore throat. These symptoms can be followed by more serious conditions including dizziness, drowsiness, altered consciousness, acute encephalitis, atypical pneumonia, severe respiratory problems, seizures. The first cases of Nipah virus infection were identified in 1998, when an outbreak of neurological and respiratory disease on pig farms in peninsular Malaysia caused 265 human cases, with 105 deaths. The virus itself was isolated the following year in 1999. This outbreak resulted in the culling of 1 million pigs. In Singapore, 11 cases, including 1 death, occurred in abattoir workers exposed to pigs imported from the affected Malaysian farms. The Nipah virus has been classified by the Centers for Disease Control and Prevention as a Category C agent. The name Nipah refers to the place, Sungai Nipah in Port Dickson, Negeri Sembilan, the source of the human case from which Nipah virus was first isolated. Nipah virus is one of several viruses identified by WHO as a likely cause of a future epidemic in a new plan developed after the Ebola epidemic for urgent research and development before and during an epidemic toward new diagnostic tests. Vaccines and medicines. The outbreak was originally mistaken for Japanese encephalitis, but physicians in the area noted that persons who had been vaccinated against Japanese encephalitis were not protected in the epidemic, and the number of cases among adults was unusual. Although these observations were recorded in the first month of the outbreak, the Ministry of Health failed to take them into account, and launched a nationwide campaign to educate people on the dangers of Japanese encephalitis and its vector, Culex mosquitoes. Symptoms of infection from the Malaysian outbreak were primarily encephalitic in humans and respiratory in pigs. Later outbreaks have caused respiratory illness in humans, increasing the likelihood of human-to-human transmission and indicating the existence of more dangerous strains of the virus. Based on seroprevalence data and virus isolations, the primary reservoir for Nipah virus was identified as pteropod fruit bats, including Pteropus vampirus, large flying fox, and Pteropus hypomelanus, small flying fox, both found in Malaysia. The transmission of Nipah virus from flying foxes to pigs is thought to be due to an increasing overlap between bat habitats and piggeries in peninsular Malaysia. At the index farm, fruit orchards were in close proximity to the piggery, allowing the spillage of urine, feces, and partially eaten fruit onto the pigs. Retrospective studies demonstrate that viral spillover into pigs may have been occurring, undetected, in Malaysia since 1996. During 1998, viral spread was aided by the transfer of infected pigs to other farms, where new outbreaks occurred. Nipah virus infection outbreaks have been reported in Malaysia, Singapore, Bangladesh, and India. The highest mortality due to Nipah virus infection has occurred in Bangladesh, where outbreaks are typically seen in winter. Nipah virus first appeared in 1998, in peninsular Malaysia in pigs and pig farmers. By mid-1999, more than 265 human cases of encephalitis, including 105 deaths, had been reported in Malaysia, and 11 cases of either encephalitis or respiratory illness with one fatality were reported in Singapore. 24. In 2001, Nipah virus was reported from Meherpur district, Bangladesh. 25, 26, and Siliguri, India. The outbreak again appeared in 2003, 2004, and 2005 in Nayagaon district, Manikanj district, Rajbari district, Faridpur district and Tengil district. 26. In Bangladesh there were also outbreaks in subsequent years. In September 2021, Nipah virus resurfaced in Kerala, India, claiming the life of a 12-year-old boy.